And now we are going to learn how to measure abundance using random sampling and the systematic sampling. We learned that random sampling with the quadrats and counting along the transects are used to obtain measures of abundance. So, what do we mean by abundance? Well, abundance is the number of individuals of a species within a given area. And the results from the quadrats or the transects can be used to calculate the predicted frequency and density of a species within an area, or to measure the abundance. Some examples include the species frequencies, species densities, and the percentage cover. Let's start by the species frequency. Species frequency is the probability that the species will be found within any quadrant in the sample area. Well, it can be calculated by the number of the quadrats that the species was present in divided by the total number of quadrats and then multiplied by 100. For example, if the bluebells were found in 18 out of 50 quadrats, then the species frequency would be 18 divided by 50, all multiplied by 100, and this would give us 36%. It can sometimes be difficult to count individual plants or organisms. When this is the case, percentage cover of the species within the quadrant can be estimated instead. In this case, the quadrant is divided into 100 smaller squares. The number of squares the species is found in is equivalent to its percentage cover in that quadrant. For example, if grass is found in 89 out of 100 squares in the quadrant, then it has a percentage cover of 89%.